Okay, good morning, everyone. Well, it was a tremendous sports day yesterday. A's and Giants won. Angels, Dodgers, and Lakers lost. Yeah. And the Warriors didn't play, so it was the ideal day. The ideal day. I just realized we only have about a week and a half before the end of the semester, and you guys start doing finals and then summer vacation, and et cetera, et cetera. I know a lot of you are getting a little notching up the adrenaline levels for the lab thing is next week, right? The lab seance? Okay. Just a couple issues. A couple of people asked some questions about it. For my part of the final, my questions, there'll be one unambiguous answer for every question. Secondly, there's no all the above, none of the above, A plus B, and every other Tuesday type questions. I don't believe, philosophically, I don't believe in those questions, those types of answers. So that's my part of the exam. In case you're wondering, if any of you guys ever end up teaching classes later on, all the stuff you hated in college, you can then do something about it, you know? It's kind of nice from that standpoint. Also, I'm setting up a review session. During sqoOhgc week, there's no class here during the normal time frame. I'm setting up a review, a question and answer review, not a review, but you guys, we'll just go over different parts. You guys can ask me a lot of questions. I think it's the Wednesday of sqoOhgc week in VLSB, but I will give you information on B-Space on that. And I'll send you out some sample questions also. Okay? So that's all coming up. Mike, anything else along those lines you want to bring up? Okay. So I will send that out by B-Space. Okay. Oh, yeah, office hours today, 11 to 12. Friday, 9:15 to 10. No Thursday office hours. You can always ask me questions by email. I've noticed the email traffic's picking up a little bit. Don't ask me questions about the lab part of it, because a lot of that stuff, very frankly, you guys know it better than I do at this moment. Certainly you will by next week. So try to pertain your questions to what we talk about in class. And the stuff I'll ask about, by the way, is only what I discuss in class. Okay? There'll be new scenarios, but it'll be based exactly on what we discussed in class. There'll be nothing from the book or the reader that might be there that I didn't quite go over. Okay? All right, so today will be our development flyby. We'll talk about cleavage reactions, which we've already talked about with humans. We'll reiterate this. Gastrulation, organogenesis, morphogenesis, and inductive signals. Inductive signals are hormone-like signals in development. Okay, a review here, quick review. Know your pregnancy hormones. Know who sends them out, who receives them, and what they do. Know your labor hormones. The main ones are oxytocin and prostaglandins, but estrogen also plays a role. There are other hormones involved. These are the key players. If you know this, when you learn the other stuff later on, it'll make more sense to you. Twins. Know the difference between a fraternal, identical, and mirror image. Okay? Two of the three, identical and mirror image, are genetically identical types of twins. Mirror image, you could be left versus right-handed. It's a non-genetic component. Creates left and right-handedness. Okay, it's an epigenetic phenomenon. And also fraternal twins, which are like siblings. Okay? Stem cells. Through mitosis, stem cells either self-renew, they're undifferentiated, or they form a differentiated type cell. That's kind of the bottom line of these things. And it's a very important issue, both scientifically as well as politically, which we got into a little bit. The only kind of stem cells that can form all your cell types, embryonic and extraembryonic, are called totipotent. In humans, the only source of totipotent cells is the zygote to the 8-cell morula prior to compaction. Okay, once it starts compacting and coming together real tightly, you start losing, the cells start having some specificity as to what they end up doing. An example of an extraembryonic tissue is the trophoblast, which is the part of the blastocyst that binds to the attaches to the endometrium lining. That is, does not form the embryo. Okay, it is an extraembryonic tissue, obviously critical. Okay? Pluripotent stem cells, which is where most of the investigation takes place these days, can differentiate probably to all embryonic tissues, but just to be safe, I said most embryonic tissues. ES cells, or embryonic stem cells, come from the inner cell mass of the blastocyst. They can, in theory, differentiate into any differentiated cell type. Two other sources of this, but that aren't really used very much, is embryonic germ cells, which are the precursors to the spermatogonia and the oogonia, and embryonic carcinoma cells. Not a good idea to start with a cancer cell, but these are pluripotent embryonic cells. These come from gonad tumors of the fetus. And then there's what's in most adults. All of you sitting there all have stem cells in most of your tissues. And you have either multipotent or unipotent stem cells. To me, they're given different names, but it's really all one and the same. And that is a restricted differentiation potential. Unipotent can go to one tissue. Multipotent can go to more than one. An example of something we'll talk about next week, bone marrow stem cells can go to the myeloid stem cells and lymphoid stem cells. So the bone marrow stem cells are multipotent. The lymphoid stem cells only go to lymphocytes. Therefore, you can consider them unipotent. Okay? Muscle stem cells only go to muscle cells. Those are also unipotent. But to me, it's all the same. They're all restricted differentiation potential. It's also a big issue in cancer cells. In tumors, there are stem cells. And if you take a tumor and you give it a certain treatment, tumor goes away mostly that you can detect. 5 years later, breast cancer, this is a very big issue, it arises back up, but with a slightly different property that's resistant to the therapy you gave it in the first place. And that is because some stem cells were there with that different property and eventually grew out of the population and formed another tumor. And that happens a lot when tumors come back. A lot of times it's not exactly the same tumor. It has a slightly different property, and that is because of the stem cells that survive the therapy. And this is a very big issue in cancer biology. Okay? Questions on that, on stem cells? And then the political/scientific issue is the fact that a lot of this can be done in vitro. You can in vitro fertilize, you get a marula, a blastocyst, you can take out the inner cell mass, put it in cell culture, take that cell culture and give it hormones, genes, other kinds of signals, and you can differentiate it into something else, like nerve cells or something, and put it back, in theory, put it back into the patient. And aspects of this are beginning to work. Okay? In my opinion, one of the very few potential for treatments of paralysis or spinal cord nerve damage is patching back into a patient's stem cells to then grow into nerve cells, forming nerve cells and patching them back in. At the moment, there's no better potential for treatment than this kind of procedure. Okay? I might be saying something different 30 years from now, but not at the moment. The issue is, obviously, an inner cell mass can make a fetus and therefore a person. So a couple ways of people thinking about this is you can take a marula. This blastocyst can make a fetus or inner cell mass and do it in vitro. In the marula, the 8-cell marula, you can take out one cell and go in vitro and take the other 7 cells. Another cell will come out of this, will divide, and you'll make a functioning fetus and embryo. So in this case, you can argue that you're not really, if you take that point of view, you're not really killing an individual because the individual is still there. You're just taking their cells. One of the things that I think you will see in the future, to some extent it's happening now, is those of you getting into medicine, is that, well, someone's going to have a baby, keep some of their stem cells around to treat something that might happen 40 years later. Okay, people do freeze down placenta and things of that sort along these same lines. So you will see this issue directly as you get into your careers and professions. It's good to think about it now. And also have something that works in primates, somatic cell nuclear transfer, and that is you can take a primary oocyte, take out the nucleus, put in a diploid nucleus, and get it to form a zygote. It works in primates, has not worked in humans yet. The issue here is when you do this, you get aneuploidy, you get extra chromosomes, which is not a good thing. So I think that's a little less, it's behind some of this other stuff, but certainly out there. And there's lots of other ways of thinking about this. You can reprogram in mice, you can reprogram skin cells to become embryonic stem cell-like in their phenotype by giving them 4 transcription factors. Okay, so there's a lot of this out there. It's a very interesting part of research, and some of you will get into this. So this is why I get into it as detailed as I do. Questions? Yes? Can you use somebody else's stem cells? In theory, you can. You have to worry about histocompatibility, right? Compatibility that your immune system doesn't recognize it. One of the ways that people think about this is adult stem cells. You don't have as many, but if in theory you can take stem cells, or your own stem cells, turn them into something else, put them back into you, you don't have to worry about that. It's definitely an issue. And there's ways of thinking about this. You can reprogram the cells themselves. Okay? The safest bet is to put your own stem cells back into you, along those lines. Not the surest thing. It's never 100%, right? But it's definitely why people think about adult stem cells. The issue there is there's not as many of them, and you have to destroy adult tissue in order to get it. Okay? All right. So let's get into development. The 3 issues in development we'll talk about, actually I have on the board there, cleavage reactions, which is a partitioning of the cytoplasm, but you have mitosis. Let me just put this over here. We'll just define our terms here real quick. So you have cleavage reactions. So it's partitioning of the cytoplasm with nuclear mitosis. Nucleus mitosis. So all the cells are diploid, just that they're smaller. So as you go from the zygote to the 8-cell morula, the total size is about the same over here. So the size of the zygote physically is the same size as the 8-cell morula, approximately, because you're partitioning the cytoplasm. And we'll talk a little bit about that. We're going to compare frogs versus humans. It's not as random of a comparison as you might think. Obviously, we want to talk about humans. We already have. But with frogs, there's a little more experiments have been done. So therefore, something a little more is known about it. And it touches on the fact that not all species do it exactly the way humans do it. So there's some interesting discussion that one can think about. And also, I can bring in my analogy in discussing frog cleavage reactions. We can bring in our analogy of Laker fans versus Warrior fans into this classroom. And you'll see what I mean here in a second. We have gastrulation, which forms the 3-layered embryo. We'll briefly talk about that. And then we'll talk about organogenesis, just from the name, forming of rudimentary organs, and morphogenesis, which is the shape. And we'll be using this classroom to discuss some of these issues, so you can see it in a 3-dimensional point of view. So it's also another term is morphogenesis, which is the shape of the organism. Okay, so cleavage reactions, partitioning of the cytoplasm. Humans, we've already discussed it in the concept of reproduction. You have the zygote, and you partition the cytoplasm to get an 8-cell morula. And then you have something that happens right after the 8-cell stage, and that is compaction. Happens right here. Compaction is where the cells come together much more tightly, much stronger cell-cell interactions. Now, thinking back all the way to about 2 weeks ago or so, can you guess as to a protein that might be involved in compaction that might be very important that we discussed? Do you know of any molecule we discussed in the last 2.5 weeks that has something to do with adhesion and cell-cell contact? Yes? Keep you in the right direction. It's not occludin, but keep going. Huh? No, those are involved in sealing. Okay, but you're in the right lecture. Okay? That's sealing, but what's involved in adhesion, adherence junctions? Yeah, cadherins. So the cadherins, which are involved in making adhesion junctions later on, are involved in keeping these cells together and plays a role in compaction. The interactions become tighter. So keep in mind, molecules that are used in development, a lot of times are used later on, in this case for cell-cell interactions. Okay? All right. The frog, however, is different. So we're going to just briefly discuss cleavage reactions in the frog, since we're all experts already on the human. Part of the reason is the frog egg is polarized. I think you guys probably had some aspects of this in the lab. You have an animal hemisphere and a vegetal hemisphere. And the vegetal hemisphere is down here, and this has the yolk. Okay? So it is polarized. And secondly, in humans, when these cells divide, all of these cells have a homogeneous mixture of components. They're all kind of mixed up, and just in any quadrant of the cell, it's the same concentration of a component. In contrast, in a frog egg, you have a heterogeneous mix of components. And that is, we can just look at 4 components, A, B, X, and Y. All the frog eggs have the same kind of general mixture, and they stay in those locations in the egg. So here's our one part of our analogy. Anybody who's a Laker fan, could you raise your hand? Okay, only if you want to admit it. Okay, so there's a few more Laker fans over here than over here. So we'll make the Laker fans, make you guys the A here, A and the X. So if the cleavage plane goes like that in this room, this cell would be Laker fan enriched versus this cell over here. So right after the first cleavage, you have the potential to differentiate into different cell types later on in the organism, just for that reason alone. Okay? How many of you follow the NBA but don't like the Lakers? I'd be in that category. See, it's a lot more enriched over here with a few over here. Over there you have agonist-antagonist interactions going on. Here you have a few more enriched in the other areas. This cell type becomes different. How many of you could care less about the NBA? See, most of you, right. You guys are the smart ones, by the way. I waste way too much time on some of this stuff. You guys are kind of spread out all over the place, right? Kind of a general mixture. And that is because you guys represent the genes that are needed for viability. Regardless whether you have an A versus B cell, Laker fan enriched, anti-Laker fan enriched, both of these cells eventually have to survive. They need cytoskeleton, they need general transcription factors, ATP, et cetera, and that stays as a homogeneous mixture. And so that's what these eggs look like on the inside. So the location of these components dictate what happens later on. Okay? So in this case, the Lakers side of the room, which was over here, right? The Lakers side of the room was over here. The playoffs hit, you guys are going to be depressed. The anti-Laker side, we're going to be happy. Now, Warrior fans sit back and watch TV and worry about the Sharks, who are down 2-1. Okay? Or actually, baseball season started, right? Anyway, the point is, the cleavage plane dictates it. So, what if the cleavage plane went this way? Instead of down the middle, what if it went this way? What we just decided was a Laker versus anti-Laker cell becomes a little different because you have different components in the cells. So if the cleavage plane was this way, so you have XY versus AB versus AX and BY. So it's a totally different potential in the cell. So frogs and similar kind of beasts do this. They have this polarity in the cell, which dictates what can happen later on. That is distinctly different from humans. So you can slice and dice these human cells all you want, and they'll be different. They're the same. That is why you can take one cell out of here and either get a totipotent stem cell, or if it happens in vivo, you can get an identical twin to the rest of the cells. because they're all the same, up to the 8-cell morula. In frogs, you can't do that. You get rid of one of these guys, and you pretty much don't get an active organism because you're missing critical components. Okay, does everybody see that difference? Okay, so what happens in frogs, this is, again, will be distinct from, you'd never see this in humans, is when you see the fertilization, but what happens after the fertilization, here's this is a little different. So it fertilizes, and what happens is the outer edge, the plasma membrane cortex, rotates towards the site of fertilization. That's the outside part, but the inside part stays the same. So the membrane, the plasma membrane, stays the same. So the way to think about this is you have a helmet on a football player, or in my case with my kids, a lacrosse player. And the helmet gets bumped. So your head stays in place, mostly. But the helmet moves. So the helmet is the plasma membrane cortex, and your head is the plasma membrane. So what happens is, when the fertilization takes place, there's a lot of reactions occur. The plasma membrane cortex moves, and you get a region of this egg, this fertilized egg now, that's called the gray crescent. And all this is an unpigmented region that for hundreds of years, as soon as they invented microscopes, even the very crude ones, you could see this. And this always happens. So why am I bringing this up? Well, in the area under the gray crescent, it occurs in a certain region of this ABXY setup, but in the area under the gray crescent, it dictates an aspect of polarity. So what does polarity mean in this case? So again, it's the components underneath. So the way to think about it is that periodic table in this room is the gray crescent. As we look at it in a microscope from outside, see, there's not as much pigmentation there. So it would say the students right here in this part of the room dictate some fate of this fertilized egg that's distinct. And that'd be like right under the gray crescent. So again, think of it three-dimensionally. It's the area underneath the gray crescent. So which polarity are we talking about here? So here's a fetus, and you have an anterior part of this, posterior. You've got a ventral side, which is the frog's belly. You have a dorsal side, which is the back. And you've got a right and a left. The area immediately underneath the gray crescent, so again, in this room, this section of students here, dictates dorsal polarity. So underneath the gray crescent, area underneath, it dictates dorsal polarity. So what does that mean? It means you're forming the back structures. And the way you can show this is what if some huge pipette comes in through the gray crescent and sucks out all the students right here? Pardon? Right, if you suck out what determines dorsal, what happens? You can't form the back, right? And you get an embryo that makes a belly only. And that's how you can show what's right underneath it is important. And that is true for other kinds of polarity, but this is a very clear one because it's under a structure that's really easy to detect by a microscope. Because you can literally stick a pipette in there, and small pipette, and suck out the determinants that are right underneath it. Okay? That's different than humans. In the first 8 cells, you get rid of a part of a cell or one of those cells, doesn't matter. You'll still form a normal, in theory, a normal fetus. Okay? Questions on that? Okay. The other thing the frog does that's different is the cleavage planes are different. Because of these determinants are so important, the cleavage planes always occur. So here's the frog. You got a zygote. The first cleavage plane is vertical. The second one is vertical. This is just, it's three-dimensional here. And the third one is horizontal. Okay? So because of this, these determinants end up in different cells. So in our analogy, we could break it down into Laker fans, anti-Laker fans, the 1 or 2 Warrior fans that are here, Knicks East Coast fans, football fans, people could care less. People are like musicals versus theater. You can break it up any way you want, but you'll have different combinations in each of these cells. And therefore, each of these cells, because they have different components, will differentiate into different structures. This is not the case in humans at the 8-cell stage, but most definitely is the case in a frog. Question?
For all, our cleavage reactions only happen 3 times for all animals?
No, they happen more. I mean, I can't give you an exact answer on that because I honestly don't know how many cleavage, but I'm bringing it up to the 8 cells because in humans, as a comparison, at the 8-cell morula, where everybody's the same, you start getting compaction, then everybody isn't the same. So at the 8-cell stage, those 8 cells are indistinguishable. Okay? At the 8-cell stage in the frog, they are not indistinguishable. They are quite distinguishable. So you get rid of one of these cells, and you end up with a different fate, or a different polarity, or a different structure later on in the organism. Okay, so at the 8-cell morula, you could pull one of them out and make another twin. Make an identical twin out of it, a complete human. Okay? Can't do that in the frogs. All right, questions on that? This is a very quickie kind of discussion of cleavage reactions.
I have a question. That you said that the frog splits at the 4-cell stage while they make a twin at that stage?
No, I did not. You can make a twin of humans because each of these cells are indistinguishable. In the frog, they are distinct. So here's our 8 cells. We got the Laker fans, the anti-Laker fans, the Warrior fans, etc. And we take the anti-Laker fans out of this, and we have a classroom in UCLA, right? So you've changed the phenotype of this. Okay? All right. Sure. Yes, one more. Yeah, do the cells communicate with each other? They are thought to, yes. Likely through gap junctions. Okay? Now, after cleavage, you have something in the human. You go to the blastocyst. In frogs, you continue with these reactions, and you get larger morullas, and they look a little funnier because the yolk side of the egg divides slower than the animal hemisphere. But eventually you get a blastula. Note in a human it's a blastocyst because it attaches, a cyst. In frogs it's a blastula. It doesn't attach because it's free swimming out there, or free moving. And the inside is also fluid called a blastocele, which is exactly the same name you see in a blastocyst. So if I say to you, what's a common structure between a blastula and a blastocyst? The answer is blastocele, because it's both a fluid, it's a generic term for a fluid containing compartment. All right? The next thing that happens is gastrulation, forming the 3-layered embryo. And this is due to cell movement and cell-cell interactions. So what happens is, here's the blastula. This is again for frogs, because humans have a slightly different point of view on this thing. And the cells on this outer part start wanting to move inward. It's as if those students in the middle of the classroom, all of a sudden the seats start moving in. Okay? And what you get is, so the cells start involuting in, and this is called a blastopore. Obviously, from the name, blastula, the pore in the blastula. And we'll just continue over here. And that process continues till you get a 3-layered embryo. So the outer part of this is called the ectoderm. The inner part, right in here, again, this is a 2-dimensional drawing of a 3-dimensional process. Endoderm. It's one word, by the way. And the middle part is called the mesoderm. The locations of these cells, and also in combination with the various determinants, dictate the final structures. So the ectoderm, an easy way to remember, is skin. Right? It's on the outside. There's other, and you don't have to memorize all the different structures that do this. Mesoderm, it's in between cells, right? One of the final tissues are muscle. And the endoderm are things like the intestines and the gut form. And you don't need to memorize that huge list of what comes out of this. So the bottom line is the location after gastrulation will help dictate, in combination with the determinants of the proteins that are there, what the final function will be. Does everybody see that? Any questions? Yes? All right, a blastocyst and a blastula. Where do you see a blastocyst? What organism that we talked about? Humans. Blastocyst, cyst. You're attaching. Where does the blastocyst attach to? Endometrium, right. It is this name of the structure, equivalent kind of structure in humans because you're attaching it. The frog gives you an example of a free-living organism out there, and it's a blastula. Fundamentally, it's the precursor for the same thing, a 3-layered embryo. Yes?
Does that mean that the blastocoil turns into the mesoderm?
I'm not sure of the answer to your question, but you only need to know it as far as I have it right here. You gave me you asked me a question that's probably more related to the lab than it is here.
Right here, the blastula looks like that.
This is the blastula. The cells on the outside, you guys are the blastula, this room is. You guys in the middle start going inward. And at the end of the process, the people that went completely inward are the endoderm. Those that migrate kind of to the in-between areas become the mesoderm, and you being on the outside of the structure is the ectoderm. So it's a physical change that dictates where these things are. You almost have to see it 3-dimensionally. Okay? Keep thinking about it. In humans, it happens a little differently. In humans, and also in birds, surprisingly, here's the structure. Instead of coming in the frog, it all kind of comes into a big pore. In humans, you have a primitive streak. Same kind of process. Cell movement, cell-cell interactions. So like a big crevice opens up, so as if an entire row is a primitive streak, and people on either sides of the row start falling into this crevice in an organized way. Yeah, in humans. Thank you. I'll reiterate. This is humans, and therefore this is a blastocyst. Whenever I say human, we're starting with a blastocyst. Frog's a blastula in the context of this lecture. Thank you. Okay? So in the blastocyst, you form a primitive streak. So it's not a little pore, but it's a whole crevice starts opening up. But you do make a 3-layered embryo out of this. Ironically, not ironically, but interestingly, birds do it this way also. They make primitive streaks. The evolutionary issue, I have no clue. Okay, but they do it this way. So in the humans, what happens is, in the blastocyst, right, you have the inner cell mass. This becomes the epiblast. And there's a hypoblast. The hypoblast is the yolk chamber, but humans do not have any yolk. But from an evolutionary standpoint, for who knows what reasons, we still have a yolk chamber. This actually, some of the initial red blood cells are produced out of here. The epiblast then forms the endoderm, the ectoderm, and the mesoderm. So if the blastocyst inner cell mass happens about day 7, this is all occurring about day 15. So about a week later. So you remember the blastocyst implants about day 7, right in the endometrium after fertilization. It takes about 7 days to make the blastocyst. It sits there, the trophoblast, you make the placenta, HCG, et cetera, et cetera, et cetera. And around day 15, you start getting the 3-layered embryo, forming the endoderm, ectoderm, mesoderm. So it's not unlike what goes in a frog, generally speaking. You got 3-layered embryo. But the structure is quite different because you're attached in the blastocyst. Easy way to remember, blastocyst, it's a cyst. It's attached to something. Easy way to remember. Okay, questions on that? I don't know how much of this you guys had in the lab or not, and I'm sure this is lab-esque light in some of the details, but I'm trying to draw a comparison and contrast to humans. Okay? Now, in organogenesis and morphogenesis, there's a couple kinds of processes that take place that can dictate structures in some unanticipated ways. So, for example, so now we're talking more about organogenesis. Okay? So one of these is cell shape, can drive some of this. So let me ask you guys a question. And excuse me, the cell shape change, cell shape can dictate this. One of these is called cell wedging. And just from the term, here's the cytoskeleton. After cell wedging, part of the cell becomes fat. Let me give you an analogy. Think about sitting in a row in this room. The constraint is the ends of the rows cannot move. What if all of your chairs, instead of being these little skinny chairs that for some of you you barely fit in them, to the nice, all you guys and your parents have these nice, easy living room chairs, these big leather chairs, right? What if every one of these chairs became one of those big, fat, wide lounge chairs? Okay, the kind of chairs you melt in over Thanksgiving, right? Just watching games all day or doing something else. Certainly not anything major. Okay, so what if every chair in this row became one of these big fat chairs, except the constraint is you cannot move outside the ends of your rows? All right, what would it look like if all of a sudden the chairs all of a sudden widened and got a lot bigger? Especially widening. What would happen to the row? And say that it's not attached to the floor. So it's free moving here. Can anybody predict what would happen if every one of these chairs became much wider? So someone say it? Yeah, it would bow out, right? Because the constraint is you can't move the ends. And that's precisely what happens. That process can drive the formation of the neural tubes, just the cell wedging. So what happens here, one way to think about it is, here's a row of cells, and you start the wedging process. So it starts bowing out. And let me just put this over here. And it really starts bowing out. Okay? And then put this over here. We don't need this anymore. We're done with cleavage reactions. So it keeps bowing out, and eventually it breaks off. So here's the original row. Here's some cells in between, and here's the part that bowed out. This becomes the neural tube. The little part that's in the middle is the neural crest cells. And what's interesting here is cells can move and migrate during development, and the neural crest cells then become bone, osteoclast, osteoblasts, etc. But what drives this structure here, does everybody see this structure here looks completely different than that structure, right? You've actually formed a rudimentary organ, a neural tube and neural crest cells, all driven by cell wedging. So it bows out, and eventually the physical dynamics wins out, and you reform the structure that looks a little different, driven by cell shape change. Okay? Now, another kind of interesting phenomena that can drive some of this is called convergent extension. So let's use the classroom once again. You have 2 rows. And the constraint is you don't change the size of your chairs. That's it. But you can move on either end. Row 7 and row 6 merge with each other to form a single layer. What would be the effect? You don't change the size of the chairs, but you can, there's no boundary, there's no constraints on either end. What would happen if row 7 and row 6 merge with each other to become a single layered row? It would get really long, right? Everybody see that if 2 rows come together. And this is another process that can help create shape, morphogenesis. Remember, morphogenesis is shape. So let's just do this with 6 cells. They start moving in towards each other. Okay? And then you get something that's much longer. That's single row. And that's by cell movement. You're not doing much except cell movement, right? And they come together, and instead of 2 rows, single cell layer, you get a much longer row of a single cell layer. That's one way you can change shape, because you don't have the constraints or the boundaries. Okay? Questions on that? And people are studying how this works. And cell-cell interactions, cell-cell contact and communication are all extremely important, as one can predict. Now, another one which we can use the classroom in is directional cues. Cells are moving all over the place, especially during development. It's kind of an interesting phenomenon. But they go and they move in certain specific paths. So you have a cell here, and it wants to go down here. What it does sometimes is it goes like this, and then goes like this to move. Why does it take this specific path? And not, at least on this blackboard, the shorter route would be from here to here, right? But why does it do this and go down? And it's because of directional cues. You have these positive directional cues here, and you have negative directional cues so it doesn't stray from the path. And a lot of this is extracellular matrix. The analogy in this classroom is, for you guys sitting in the middle, you're coming in the top of the room, right? What's the shortest path? Not figuring out, difficulty of getting you these seats in the middle, right? You come in the room and you walk over the tops of the chairs to the middle, right? Isn't that the shortest path, right? But instead, what do? You walk down the aisle and then you come in, right? Walking down the aisle and coming in is your positive cues. There's nothing in front of you, you can't trip, right? And so forth. You walk on top of the chairs, you have all sorts of negative cues. It's hard to balance yourself. If I tried it, I'd probably break my neck about the second row of trying to do this, okay? So you have all these negative cues. Even though the distance is not the shortest, the positive cues tells you how to go. Same thing here, except it's on a molecular level, and that is matrix protein signaling and so forth tells these cells exactly where to go and how far to go. And all this is going on during development. This creates morphogenesis and organogenesis. So these are just a couple issues that you'll see in more detail later on, those of you who get into developmental biology later on. This is just a little taste of some interesting phenomena that one sees. Questions? Okay. Now, the last little bit that we will continue with on Friday, and that is inductive signals. What drives some of this stuff? It's given a term called inductive signals. You're inducing processes in development. But what they are in reality is hormones binding to receptors and getting a response. I mean, that's really what inductive signals are, or hormone-like phenomena, I should say. I'll just down here. And as you can imagine, there's two general classes of inductive signals. You've got positive and you have negative inductive signals. And let me just give you the generic viewpoint on each of these. And this is just with circles and squares. So here you have two cells sitting next to each other. These are cells. When you have no signal around, these cells are round. Okay? This is by analogy, in case you're wondering. There's no absolutely round cells that don't require a signal. Okay? A positive inductive signal comes in from one of the cells. So this cell now starts making a positive signal. Goes to the neighboring cell and turns it square due to interaction with the receptor. So the square cell requires a signal. Whoop. Whereas the round cell is no signal. So what a positive cue, a positive inductive signal is doing is saying, here's a cell type that doesn't have a signal for the moment, and I'm going to give it a signal and now turn it into something else. In this case, a square cell. Does everybody see that? Should be pretty straightforward. What's also around is very important are negative signals. So in this case, again, you have two round cells next to each other, but you need some sort of positive signal to stay round. So you need a positive signal to be round. And then what happens, a negative signal comes in and stops the action of the positive signal. So in this case, here's the positive signal coming out, but it's stopped by a negative signal. And you could say, yeah, that's an antagonist, and that would be correct. And as a result of having no signal, the cell now turns square. So in this case, if you inhibit the positive signal, you get the square cell. And you have to see operationally the difference here. In the top one, the presence of a positive signal gives you a phenotype. In a negative signal, an inhibition of a positive signal gives you a phenotype. And those are two different types of signal transduction. Questions? Yes?
I just want to double check. Is that a negative sign in the little bubble, or is that a...
Right here? Because this is this positive signal. Thank you. What happens is it's trying to tell its neighboring cell to get round. And the negative signal comes in and blocks it. Okay? Questions on the... We're not going to get into the specifics till Friday. Remember I said to you under the great crescent. What did we say the polarity was under the great crescent? Dorsal, right? Well, it turns out that signal is a classic negative signal that's under the great crescent that leads later on to the back. And that's what we'll discuss as we finish this up and get into our systems flyby. Okay, have a good rest of a day or two. I remember I have office hours 11 to 12 today. And for those of you that are not there, I'll see you Friday.